Forget about the hyphen and it becomes dactinomycin. Okay? So, one and the same thing. Then anthracyclines are a group of compounds which are exemplified by doxorubicin and its sister compounds. Doxorubicin is the prototype. Bleomycin, part of your drug list. Mitomycin is not. So you have these members to concentrate on, and the tococyte and the antimitotic drugs. The fifth one is the fifth group is the only member in this is enzyme. It's an enzyme group, and the only member so far is L-asparaginase. You know that asparagine or any other amino acid, they exist either in the D or in L form. So for the humans, okay, it's the L form which is important, okay, rather than, and for the carbohydrate it's the D form, okay. So for the amino acid it's L form, okay. So L-asparagine is an important amino acid essential for the cellular growth and metabolism. So L-asparagine can be cleaved down, okay, by an enzyme which is called L-asparaginase. Okay, so if it is in the circulation, there is no L-asparagine, then the fast-growing tumor cells, they are deprived of this essential amino acid, and they are gone. Okay, so they lyse up. Okay, so the second group, as someone was asking about, which other members? So one is there are few groups, okay, and we have examples against each. So GnRH is the gonadotropin-releasing hormone analog, leuprolide, part of your drug list. Estrogens also, okay, but we won't focus much on them. Anti-estrogens are more important. So there are tamoxifen, for example, and other sister compounds of tamoxifen, but for your drug list is tamoxifen. Progestins, androgens, and anti-androgens, okay? The anti-androgen flutamide is more important than the androgen itself, okay? Part of your drug list. So adrenocortical steroids, prednisone, we already discussed the role in certain cancers of blood cells and lymphocytes. So leukemias and lymphomas, and also sometimes in others. Adrenocortical suppressants, okay, for certain hormone-producing tumors. Not that one.
Yeah.
And the immunoglobulins and tyrosine kinase inhibitors. Okay, so immunoglobulins and tyrosine kinase inhibitors. And I limited my list to these four, which are part of your drug list. There are more as well. But remember these four, they are more important. So imatinib, the prototype, then trastuzumab, the famous Herceptin, market name, then cetuximab, okay, then bevacizumab, okay. So all of them are part of your drug list. And from the miscellaneous, remember I told you that there is a sixth group in the alkylating, when we were discussing alkylating agents. We listed 5 groups, and we mentioned there is a sixth group. Yeah, so cisplatin is actually, and its sister compounds, they are the sixth group. They are not classical, part of the classical alkylating agents, but they work pretty the same way. We will see in the next lecture how. Now, this is, I hope now you realize that there are a number of drugs, a long list of drugs, and we would focus on a few of them which are more important in terms of frequency of use, okay, or some important aspect, okay, for understanding of the theory behind it. So the cell cycle specificity of the anti-cancer drug is the focus in this slide. Could you suggest what it is? What this? Okay, so this is a normal cell cycle. Okay, rather phases of normal cell cycle. What is G naught?
G naught is the phase where
This
the cells
one,
which
the green
do not
G naught.
divide. Which are not divided.
G naught or G0 is the one in which the cell is not dividing. It is functional.
They have the capability
Okay?
to divide.
For example, a functional neuron, a functional myocardial cell. Okay? Okay? So they are functional, okay, but they are not an active division. Okay? If a cell starts cell division, the first phase is G1, or the first growth phase. Okay? First growth phase. So what is G?
Growth.
Yeah. So what is G1?
Protein synthesis. No. It's
DNA goes to S phase. Organelles in the G2, isn't it? So the machinery required for DNA replication. Okay. So all the enzyme pathways which are required for DNA replication, they are the ones being focused in G1. Okay. Then comes the S phase or synthetic phase, synthesis for DNA. Okay. And in this phase, the two chromated chromosomes, they become 4 chromated chromosomes. So the number of the chromosomes is still the same, but the content is doubled. Okay? This is followed by G2 or second phase of growth, second growth phase, which is characterized by, the organelles, they catch up the demand, they are duplicated and they multiply to meet the demands in the daughter cells. And then, once everything is ready, then there is active cell division. Okay, the M phase, the mitosis phase. Okay, so mitosis is exemplified by formation of a mitotic spindle, which divides the chromosomes into 2, and then each daughter cell receives exactly the same number of chromosomes. Okay, that's what you already know. And these are the checkpoints where certain proteins and associated factors, they can act to favor entry into the next phase. Okay? So the most important are the cyclins from the cell cycle. Cyclins. Okay? So cyclin group of proteins and enzymes, they work in synchrony to facilitate transition from one phase to the other. These are the checkpoints. Okay? Okay? Done?
Okay.
Then stamp the visa and let the cell go into the next phase. Okay? So, we have drugs which can work in all phases, and we have drugs which can work in some phases. Okay? So, based on that, there is a cell cycle non-specific group of drugs. Now, this is a different classification. Respecting the cell cycle specificity. Okay? So, cell cycle non-specific drugs are the ones which can damage the cell, okay, in all phases. Right? So, whether it's in G0, G1, S, G2, or mitotic, so all phases it's effective. Louis? Okay, so cell cycle non-specific drugs are exemplified by the horrendous alkylating agents, okay, and we will see how they work. Yeah? There are others as well. The cell cycle specific drugs are the ones which are targeting the cells which are either actively dividing or actively metabolizing. Okay? So what happens during active metabolism? There is a lot of gene coding and coding, protein synthesis going on. So whenever the DNA is active, okay, is in replication state, those drugs will be effective. So cell cycle specific, okay, acts during active cell cycle, but very minimal during G0.
All except GC.
Sorry?
All except GC.
Yeah, you can say that. Okay, examples are shown. Then phase specific, of course, they would be effective during the active cell cycle, but not in all phases. In one or two specific phases. Let's see. One of them is, what is this? Hydroxy arabinoside. It's a purine analog. Yeah?
That's Arab in nature.
Okay? Logically, should be effective in S phase. Similarly, another purine. Okay? And antifolate. Then you have the paclitaxel and vinca alkaloids targeting the M phase. Okay? Now let's see the subject from the third perspective. Okay? The third perspective is site of action. Okay? So the first we discussed cell cycle specificity, and now the third perspective is site of action. Which site actually? Okay? So you can see, gentlemen, be gentlemen. Okay? Okay, be gentlemen. Pay attention. And have a look. Otherwise, you will be roaming around crying, after the people that we didn't understand that one or this one, and etc., etc.
As long as it's
Who is at loss ultimately? Not me. It's you. So do yourself a favor. Pay attention. Okay. This is the last warning.
I have a question, doctor.
I will just purge what I'm supposed to, and I'll go away. Okay. It's you who have to appear in exams, who have to excel in your features. Okay. So if you are not attentive, you will be the loser. Not me.
As long as it's in the slides, we'll be okay.
Yeah.
No, but the exam's not fair. How can you judge us?
So you have to be physically and mentally present. Okay. Not like just physically. You have to be mentally prepared to take the dose. Okay.
But also the exam shouldn't be focused on only one area. Like a lot to question.
Sure.
So you can't judge us on that.
So let's see. Let's see. This is a typical animal cell, okay, and this is a supporting or feeding blood vessel, okay. Now, could you identify the organelles? I'll put the arrowhead. What is this? Mitochondria. What is this?
Gold endoplasmic.
Rough endoplasmic reticulum.
Yes.
What is this? Smooth endoplasmic. Yeah. Not that. Microtubule. Okay. What is this?
Lysosomes. What's it called?
Okay. So you know it. Okay, good. You remember it. Very good. So let's see. What target we have in blood.
Centrioles.
What target we have in blood? Let's start from periphery towards the center. So what target we have in blood? Circulating amino acid L-asparagine. Okay? So asparagine is in circulation. Immunoglobulins and hormones at their own receptors. Okay? So membrane receptors, okay? And cytoplasm, whatever. Vinca alkaloids, microtubules. So it could be other microtubules as well. You know microtubules. They are important in intracellular transportation. Yeah, so they are molecular motors actually. Antimetabolites are targeting a number of processes